Hi, Bio 47. I'm going to be going through Lab 16, the second part, all about the respiratory system. This starts on page 5 of Lab 16 in your lab manual. So we'll be going through the gross and microanatomy of the respiratory system. The first part is the upper respiratory structures. These are all that are residing outside of the thoracic cavity. The first part is the nose and the nasal cavities. They begin the preparation of inhaled air for the respiratory pathways. And the air, as it's passing through the conduction zone, is being cleaned, warmed, humidified. So it comes in through the external nares. Those are the nostrils, the opening into the nose. It's guarded by hair. And next we would see the nasal septum. We can't see it on this view, so it's not exactly a mid-sagittal view. The nasal septum is what separates the two sides of the nasal cavity. It's formed by the perpendicular plate of the ethmoid bone and the vomer bone, as well as some cartilage. What we can see are nasal turbinates or nasal conchae. Remember, that's the superior, middle, and inferior nasal conchae. They're made by the ethmoid bones, superior and middle nasal turbinates, and the inferior nasal turbinate. Also visible in this section is the hard palate. The hard palate is the anterior floor of the nasal cavity. It separates the oral and the nasal cavities. It's made of the maxilla and the palatine bones. And this posterior floor of the nasal cavity is the soft palate. That's separating the oral cavity from the nasal cavity. And this part that hangs down from the soft palate, remember, that's the uvula. Also visible here are some of the paranasal sinuses. These are mucous membrane-lined cavities within the frontal, ethmoid, sphenoid, and maxillary bones. They function in sound production, air moistening, and reducing skull weight. We've already talked through the pharynx, which we talked about in the context of the digestive system, which number 1 here would be the nasopharynx, number 2, the oropharynx, and number 3, the laryngopharynx. This is a cadaver view of those same structures that we were just looking at, and this view is a mid-sagittal view, so what you can see here is the nasal septum. But if we take a slightly parasagittal view, then we'll be able to see the openings into the nose, the nares. We can see those nasal turbinates. We can make out the hard palate and the soft palate with the uvula hanging down, and the nasopharynx, oropharynx, and laryngopharynx. Next, we'll move into the larynx. We're now into the lower respiratory system. The larynx, or the voice box, is that large cartilaginous structure at the top of the trachea, and it guards the opening into the lower respiratory structure. The larynx is supported by 9 total cartilages, and these cartilages function to help keep open the respiratory pathway, to help ensure breathing, to help adjust the tension of the vocal structures for sound production, and to protect the respiratory pathways from the entrance of food. So this one right here, this big shield-shaped one, that's the thyroid cartilage, or the Adam's apple. There's just one of these. It's the largest larynx cartilage that forms the majority of the anterior and the lateral surfaces of the larynx. Inferior to that is the singular cricoid cartilage. It's the only complete ring of cartilage in the entire respiratory system, and it's located immediately inferior to the thyroid cartilage. You can also see the epiglottis, that sort of spoon or horseshoe-shaped elastic cartilage that projects superior to the glottis. The glottis is the opening into the larynx, and it functions to cover the glottis during swallowing of food, and that prevents ingested particles from entering into the respiratory system. Also present are the arytenoid cartilages and the corniculate cartilages. And both of these are functioning to regulate tension on the vocal cords and affect vocal tones. The bigger ones, these paired pyramid-shaped cartilages on the posterior superior edge of the cricoid, those are the arytenoid cartilages. And then the horn-shaped cartilages on top of them are the corniculate cartilages. Next, we get into the vocal folds, the sound-producing structures, the true and the false vocal folds, or the vocal cords and vestibular vocal folds. The true vocal folds are highly elastic ligaments that are covered by mucous membrane. They stretch from the thyroid cartilage to the arytenoid cartilage, and they project over the opening of the glottis. The vibrations that are caused by air flowing over them functions to produce sounds. So they're the true vocal folds. Superior to the true vocal folds are those false or vestibular vocal folds. They're relatively inelastic. They're covered with mucous membranes. They're superior to the true vocal folds, and they help to prevent foreign objects from entering into the larynx and to protect those delicate vocal folds. Then we move down into the trachea. The trachea, or the windpipe, is the tough, flexible, always open tube that connects the larynx to the primary bronchi. It's about 1 inch in diameter and about 6 inches in length. It passes posterior to the esophagus. It's lined with pseudostratified columnar epithelium that helps to condition the air. It is held open by these C-shaped rings of hyaline cartilage that keep it always open. The posterior aspect, where there isn't any of that hyaline cartilage, has the trachealis muscle, and that connects the ends of the incomplete rings. It contracts during coughing to decrease the size of the trachea and to force air more quickly through the trachea. At the carina, this last ring of cartilage at the end of the trachea, that's where we get the split into the primary bronchi. So the primary bronchi branch from the trachea at the level of thoracic vertebrae 5, and the ridge called the carina marks the division. This ring of cartilage right here, that would be the carina, and then we would be in the left and the right primary bronchi. Both the left and the right bronchi, they're supported with C-shaped rings of cartilage. They're lined with ciliated epithelium, and they contain some smooth muscle. The primary bronchi enter into the lung tissue at a groove that's called the hilus. So this groove in the lung, where the bronchi enter, that's called the hilus. And from there, they branch into secondary or lobar bronchi. So each primary bronchus divides into, on the right, 3 lobar bronchi, and on the left, 2 lobar bronchi. These are secondary bronchi that enter into each of the lobes of the lungs. They're supported with cartilage. They contain smooth muscle. And those further branch into the tertiary or segmental bronchi. These are supported with cartilage, and they contain smooth muscle as well. So up here, that primary bronchus entering into the lung at the hilus, and then branching into secondary or lobar bronchi, and branching into tertiary or segmental bronchi. These continue to branch and continue to just conduct air and branch into bronchioles and terminal bronchioles, and these are the last of the conducting passages. The bronchioles are distinct from the bronchi because they have no cartilage support, and they're dominated by smooth muscle. These branch to form the respiratory bronchioles. They're the narrowest lung passages, and they're connected to the alveolar sacs, those clusters of air sacs where gas exchange occurs. Respiratory bronchioles don't have any cartilage, they don't have cilia, they don't have mucus cells, but they do have high concentrations of elastic fibers and some smooth muscle. Then we're into the alveoli, or the air sacs, the tiny hollow alveolar spheres that are lined with simple squamous epithelium. So alveoli are organized into clusters called alveolar sacs, and each one of them has a central alveolar duct that links them to the respiratory bronchiole. And here you can see this transition from the terminal bronchiole that has a low cuboidal epithelium to the respiratory bronchioles that have that simple squamous epithelium, as well as some alveoli along the walls of the respiratory bronchioles. So now we're getting into the respiratory zone and leaving the conducting zone as soon as there start to be alveoli in the walls. And then we have alveoli here, the individual alveoli where that gas exchange is occurring. So that very thin layer that's separating air from blood. So if you see something like this that looks sort of like grapes, this is the histology of the alveoli within the lungs. Then the lungs, we're looking at gross anatomy. They're the structures that are composed of the conducting passageway, the alveoli, and associated vascular tissue. The left and the right lungs are located in separate pleural cavities. Remember that each one is surrounded by a pleural membrane. There's a visceral layer around the lung and a parietal layer along the thoracic cavity. This is a serous membrane, and it helps to keep the lungs inflated. Each lung is shaped sort of like a blunt cone, with the apex up here, just above the clavicle and first rib, and the base resting on the diaphragm. The right lung is larger, and it has 3 lobes. It has a superior lobe, a middle lobe, and an inferior lobe, separated by the horizontal and the oblique fissures. The left lung is separated into 2 lobes by an oblique fissure. So it's only 2 lobes. It has a distinct cardiac notch, which accommodates that leftward displacement of the heart. The right lung is shorter, and it's more broad than the left lung, which is a little bit longer. Here we see them in cadaver view. So in the center here is the heart, surrounded by the pericardium, its own serous membrane, and then we can see the left and the right lung. And what's been peeled away here is the pleural membrane. So this would be the pleural membrane that would be wrapping around these lungs. And there's the horizontal and oblique fissure of the right lung. And you can sort of see the oblique fissure over here of the left lung. And there's the cardiac notch of the left lung. And in this next image, the heart's been cut away. We have a frontal section through the lungs so that you are able to see the hilus, that indentation in the lung where the primary bronchi is entering and where the pulmonary artery is entering and pulmonary vein is exiting. So that's it for lab 16. Check out the corresponding visible body exercises and make sure that you familiarize yourself with the histology of lung tissue. It's not too difficult. What you'll be looking for are those spheres of simple squamous epithelium that indicate the alveoli. And let me know if you have any questions.